Programmed cell death is a process in development and in many disease pathways. Throughout the life of many organisms, many cells will die through programmed cell death. While the most well-known type of cell death, apoptosis, has been well studied and characterized, many of the other types of cell death remain unknown, and much about their processes is still unknown. Our lab studies phagoptosis, a non-autonomous form of cell death. In phagoptosis, one cell murders another cell and engulfs it. Our lab is using Drosophila as a model system to study phagoptosis. In phagoptosis, cells use eat me and don't eat me signals to induce death. Although somewhat characterized, much about the proteins involved in this process remains unknown. During oogenesis in the Drosophila ovary, follicle cells murder and engulf nurse cells in late-stage egg chambers, and the nurse cells provide the egg with nutrition. Through learning more about the proteins involved when the follicle cells murder the nurse cells, we can learn more about this new type of cell death. Previously in our lab, a proteomics experiment was done to tag the proteins that were being secreted by follicle cells during the time of nurse cell death. To determine which of these proteins is actually important in programmed cell death, we knock down the genes using RNA, interfering RNA, to determine when these genes are knocked down, if it will impact cell death. We found that these genes, none of them had a significant impact on cell death in late-stage egg chambers. However, two of the genes, Cruppel homolog 2 and thick veins, caused mid-stage death, and the nurse cells in mid-stage egg chambers were dying, indicating that these genes may be involved in preventing and regulating cell death. Abnormalities in the regulation of cell death are involved in many disease pathways, so this finding is very exciting. Too much cell death is involved in diseases such as degenerative disorders and immunodeficiency diseases, and too little cell death is involved in cancer and autoimmune disorders. Through understanding more about the proteins involved in regulating cell death and the impact of losing these proteins, we can better understand the factors that are contributing to these diseases.